URL up here. And this gives you a little bit of information and gives you the link for the application as towards the bottom of the page. There we go. As you can see, it's $2,000 a semester for 4 semesters. Okay, so it's worth $8,000 total over the presumably 2 years we will spend up at University Park. You need to meet the entrance to major requirements. If you don't know what those are, click on that link. And the change of campus requirements, also clickable. The selection criteria are fluid at the moment because apparently they have 1 application so far. The deadline is the 15th, I think, yes. So once they see the pool that they have to choose from, they'll create the selection criteria at that point. So this is only, so if you're staying for Abington, this doesn't apply. This is only for change of assignment, change of location students, and you have to remain in Everly to keep this. So if you get up there as a bio major and then you decide, oh, I'm going over to kinesiology, you can't take Everly's money to the other college. But if you remain in Everly, you get the money. So if you're changing assignment up to Park next fall, this might be something to. I didn't click on the application link, so I don't know what the application asks for. But for $8,000 is probably worth a few minutes of your time to apply. Okay, so deadline is you have a little bit of time, but don't forget about it. Okay, so that's available if you're transitioning up to University Park. Okay. Just a quicker question. We haven't done these in a while. Okay. Where in the cell does translation take place? Nucleus, cytoplasm, aluminum, FDR, Golgi, or lysosomes? Okay, we already say A in the nucleus. Of course, it's the cytoplasm. Right? Stuff has to leave. The messenger RNA leaves the nucleus, goes out into the cytoplasm, hops onto a ribosome, and is translated in the cytoplasm. Part of the transfer RNA that base pairs with the messenger RNA is called the what? Codon, anticodon, D-loop, Scheindel's RNA sequence, or none of the above. Down to 2, that's good, I guess. Of course, it's the anticodon. Right? The triplets in the messenger RNA are the codons, so the part on the messenger RNA is the anticodon. Into which site on the ribosome do charged transfer RNAs enter? E, the P, the A, any of them, or none of them? Site. lysyl site. Which direction do ribosomes read the messenger RNA? 5 to 3, 3 to 5, either way or neither way? True or false? Translation requires an array of protein factors in addition to ribosomes. Excellent. Indeed true. Initiation factors, elongation factors, release factors. Okay. Other than that last one, I think we have identified an array of pieces of information about translation that you guys seem to need to go over. So that's good. That's what these are all about. So I encourage you to look at translation. Again, I hope again, because if you don't understand translation, I don't think you're going to be able to understand this next topic. Because, crazy me, I keep fielding topics on top of previous topics. So, but we're constrained with our time, so whether you understand translation or not, we have to move on. So we took a little sidestep on Wednesday to look at one type of control mechanism that the cells have to control which genes are on or off, and therefore what proteins are made. Now we're going to get back to those proteins that we were making on those ribosomes and follow some proteins through what's called the endomembrane system. That's composed of the endoplasmic reticulum, the Golgi, and the lysosomes, as we see in this first figure. So we'll see that there are two different kinds of endoplasmic reticulum. There is the rough endoplasmic reticulum over here that's closest to the nucleus. And as you can kind of see, is composed of sort of flattened little sacs that have ribosomes attached. So we'll see shortly that the rough ER is going to be involved in producing the set of proteins that have to go through this endomembrane system, and we'll talk about which proteins do and do not have to go through that system. But they'll start here at the rough ER. You can see attached to it, but usually a little bit further from the nucleus, is the other type of ER called the smooth ER, the smooth endoplasmic reticulum. And you can see that the shape changes pretty dramatically from sort of flattened sacs to a much more tubular shape. If shape changes, probably what else changes? Function, right? So we'll see the smooth ER is going to have a very, very different function than the rough ER. Those proteins that we're going to spend most of our time following will start here at the rough ER and then travel to this next organelle called the Golgi. Golgi, just Golgi or Golgi apparatus or Golgi complex, whatever. Another series of sort of flattened sacs where the proteins that travel here will be modified and sorted to their final destination. Sometimes that destination is outside the cell, it gets secreted, but sometimes some of those proteins are going to go to small vesicles called lysosomes, which if you remember from chapter 6, I believe, in 110, lysosomes are sort of the digestive system of the cell. They contain enzymes that will break down other biological molecules. Okay, so the ER, the Golgi, and lysosomes make up this endomembrane system, as it's usually called. So kind of just work our way through these three organelles. So we'll start out with the endoplasmic reticulum, usually abbreviated ER, just for simplicity. And these are transmission electron micrographs of the two types. Okay, so remember how that works. We slice through and we can see high magnification, high resolution images of the sort of internal parts of whatever we're looking at here. So again, on the left here is the rough ER. And you can again see is sort of this series of little flattened sacs, all these little spots on them. Those spots again are ribosomes, and they give a rough appearance under the microscope, so that's why they're called rough ER. All the ribosomes give it sort of a rough appearance. And on the right we see a cross-section through all those sort of tubular structures here. So if you kind of slice through those tubes, you kind of see all those sort of little cross-sections here of those tubular parts with a smooth ER. So again, basically all of these kinds of drawings here are based on micrographs and microscopic evidence like this. So I guess before we get into what's kind of happening and exactly how we get proteins to this endomembrane system, again, the ER, the Golgi, and lysosomes, probably better to start here on this right-hand side, because that really is where we pick up from our translational stuff that we were talking about. So we kind of talked about this sort of general translational idea that's shown here. So we saw ribosomes hooking up with messenger RNAs and transfer RNAs, and we make the protein. Right? That's what we finished up with on Monday. And if we kind of treat it just like we did on Monday, where these ribosomes are just floating around the cytoplasm, then the general sort of types of proteins that are going to come off these, what are usually called cytosolic ribosomes, ones that are just floating around the cytoplasm, are proteins that are either going to remain in the cytoplasm. So, for example, say all of the enzymes for glycolysis. Right? You want to link it back to that example. Right? All of that happens in the cytoplasm. So all 10 enzymes of glycolysis and the 1 or 2 enzymes for, say, fermentation, all would be produced by ribosomes that are just kind of floating around the cytoplasm, and those finished enzymes will remain in the cytoplasm, right? Because that's where glycolysis and fermentation take place. But also off of those ribosomes will come proteins that go into some other specific organelles. So we've already actually looked at this kind of indirectly earlier. So if the protein needs to go to the nucleus, we've already studied how that happens, right, through nuclear pores and interactions with nuclear transport proteins and stuff like that. Those proteins that need to get into the nucleus, so think RNA polymerase, DNA polymerase, all the ribosomal proteins are good examples, would also be made on these ribosomes that are, again, just floating around the cytoplasm. And if they need to get to the nucleus, what do you need to do? What do you need to have, I guess, to get into the nucleus? What do we call that? What was the one?
Entry signal.
I can't hear when you whisper.
The important protein.
Pardon me?
Important protein.
That's right. We need that to interact with the protein, but how does the important protein know to interact with, say, a ribosomal protein or DNA polymerase?
The signal protein.
Right. There's a nuclear localization signal. There is a signal of some kind. That is going to be one of our big themes in this topic. So there was a specific signal that got you to the nucleus. We call that the NLS, right? The 4 to 8 positively charged amino acids. So that was our first kind of signal that got you to the nucleus. Well, it turns out that there are other signals that other proteins have to get them to other places like the mitochondria. Okay, so think about all the enzymes inside the mitochondria, many of which are made by genes in the nucleus, need to get to the mitochondria. So there's a signal for that. We're not going to worry about what that specific signal is, but there is a separate mitochondrial signal. If we're in a plant cell, there is a separate chloroplast signal. Okay, so think about, say, the Calvin cycle proteins or something like that. We'd need to have a signal to say, hey, I need to get to the chloroplast. And there are signals for proteins to get into things called peroxisomes. We're not going to study peroxisomes. A couple of topics we just have to kind of skip over because we just don't have time. But the overall idea here is that there are specific signals to get proteins to specific locations. That's going to be our theme, because we're going to see the same thing here. If you have to get to the ER, there's going to be an ER signal, and that we are going to study shortly. Okay, but so if you have a specific signal to a specific organelle, then you're going to go to that specific organelle. Where do you think you go if you have no signal at all? Right, you stay in the cytoplasm. So the default is you stay in the cytoplasm. And again, how do we know that? Through simple experiments where, for example, if we have a ribosomal protein that has a nuclear localization signal, and we remove the nuclear localization signal, that protein stays in the stays in the cytoplasm. Okay, so the experiments for that are pretty straightforward. So I think I have an explanation for this.
All nuclear encoded mRNAs are transported. Special targeting sequences direct the protein to a specific membrane or organelle. All nuclear encoded mRNAs are transported through nuclear pores and then translated in the cytosol. Special targeting sequences direct the protein to a specific membrane or organelle. For example, nuclear proteins fold in the cytosol and then enter the nucleus through the nuclear pores. Peroxisomal proteins also fold in the cytosol and then cross the peroxisome membrane into the organelle lumen. Special targeting sequences direct mitochondrial proteins to where the inner and outer membranes are close together. These sequences are cleaved in the matrix. Some proteins are incorporated into the inner membrane. Other mitochondrial proteins cross into the inner membrane space, while others remain in the matrix.
So there must be other signals to do that.
Chloroplast proteins are processed in a similar way. They cross the outer and inner membranes to enter the stroma. Some fold in the stroma and remain there. Other proteins cross the chloroplast membranes, transit the stroma, and fold in the thylakoids.
So all of these different locations presumably have different signals associated with them so that a protein gets to its correct place. Because if that protein that's supposed to be in the stroma gets inserted into the membrane, then obviously it's not in the wrong place and won't be able to do its job, and vice versa. If that protein is supposed to be in the membrane and is just floating around the stroma, it's probably not going to do its job correctly. So the idea that there are targeting signals or other types of signals to get our proteins to the right location is a really important one. So I think it's just a general one for the endomembrane system, so let's take a look at that too.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
So one type of protein that we're going to follow in this topic is a protein that's going to end up being secreted from the cell. So I think insulin is a good example of that. Okay, so if you're insulin, you have to travel through the rough ER, through the Golgi, come out the Golgi, and be secreted. So that's one type of protein we're going to follow. We're also going to see proteins that are going to be inserted into the cell membrane. So think back all the way to probably September when we covered that topic. Now we're going to see how that happens, how do they get inserted, and then how do they get to the cell membrane. And then other proteins will be lysosomal enzymes, so we'll follow those as well. So those are sort of going to be our big three types of protein that we're going to look at here in this topic.
The signal sequence of a membrane protein directs the ribosome to the rough ER. The growing polypeptide chain enters the lumen of the ER through a protein import channel in the membrane. There, the signal sequence is cleaved, sugars are added, and the protein folds. Some enzymes or structural proteins remain in the ER. Most proteins that are soluble in the ER are transported in vesicles to the cis Golgi. In the cisternal maturation model, the cis Golgi matures to become the medial Golgi. A new cis Golgi is formed by fusion of ER vesicles. The maturation process includes retrograde vesicular transport of resident Golgi proteins. Proteins destined for secretion after modification in the Golgi are transported from the trans Golgi to the plasma membrane. When the membranes fuse, the proteins are released into the extracellular space. Soluble proteins on the outside of a cell can re-enter by endocytosis and are sorted to lysosomes for degradation. Other proteins, including lysosomal enzymes, sort directly to lysosomes from the trans Golgi.
So this is basically what we're going to be looking at in this topic over the next few classes. Okay. So in this topic we're going to focus, as I've been saying, on this sort of left-hand side. So we're going to focus on these three major types of protein. So proteins that are going to be secreted from the cell, again like insulin, lysosomal enzymes that are going to end up in these special vesicles called lysosomes, and proteins that are embedded in the membrane that will eventually be, say, we've seen translocation kinds of proteins, right? Facilitated diffusion proteins, things like that that we've already studied from, sort of a functional point of view and a structural point of view. Now we'll, again, sort of finish that part of the story, and we'll figure out how they get into the membrane and how they get from the ER through the Golgi and to the plasma membrane. So we're going to trace those three types of proteins here. Okay. So if you're one of these three kinds of proteins, okay, so if you're a protein that's going to be secreted, a lysosomal enzyme, or a protein that needs to get inserted into the membrane, you're going to have a special signal. Okay, just like if you were a protein that needed to get to the nucleus, you had a specific signal. So, special signal here to get to the ER consists of 16 to 20 hydrophobic amino acids at the N-terminus of the protein. So, if you remember, we make proteins from the N-terminus to the C-terminus, so this is basically like the first 16 to 20 amino acids of the protein. Okay, so here we are at the start, so here's our messenger RNA, here's our ribosome. So the story always starts with ribosomes that are just floating around in the cytoplasm. But if this particular protein that's coming off here, the red part here, is one of these three types of proteins, again, something that's going to be secreted, or a lysosomal enzyme, or a protein that needs to get into the membrane, the first 16 to 20 amino acids are all going to be hydrophobic. So it's more this chemical idea than specific amino acids. So any of the amino acids that are hydrophobic that we looked at back in September would be in this category. So it's not just just say glycine. It's glycine and alanine and all those hydrophobic ones. So their exact identities don't seem to matter. It's just this sort of long stretch of hydrophobic amino acids. So that's what they're representing here in red. Okay, so that's the signal sequence, specifically for the ER. Well, as usual, if just having a signal is not enough, right? You need the signal and something that kind of recognizes the signal. Think back again to how we get proteins into the nucleus, right? It's not just the NLS, it's that important protein that sees that sequence, hooks up with it, and lets it go in. Sort of the same idea here. So something needs to sort of see that signal sequence. And what sees that signal sequence is this little green protein here in this picture, abbreviated the SRP, which stands for signal recognition particle. Which is an interesting little protein. Well, actually, it's several proteins. It's 6 different proteins plus a piece of RNA. So I've kind of lost track now of how many times we've seen this kind of phenomenon. So hopefully we're getting used to seeing combinations of protein and RNA together. Okay, so here's another example of that. So this little signal recognition particle, again, 6 different proteins and a piece of RNA in there. And what it does, you can kind of see here, it recognizes that signal sequence, a stretch of 16 to 20 hydrophobic amino acids, and binds to it. And in some way that we don't have all the details for yet, somehow interacts with the ribosome as well and stops translation. Not permanently, but as we'll see, but while it's attached. So it's attached to the protein that's coming off and sort of attached to the ribosome. It sort of tells the ribosome, okay, stop translating for just a little bit. Okay, so far so good? Okay. So now let's think on the other side. So if this messenger RNA was coding for something that stays in the cytoplasm, this doesn't apply, right? Because that protein would not have this signal, and therefore this SRP would not bind. Okay, so that would just stay in the cytoplasm. Okay, but if you have this signal, the SRP binds to it, and what this SRP is going to do is get this whole thing, this ribosome with the little protein coming off and the messenger RNA, to the rough ER. So how does it do that? Well, it just so happens on the cytoplasmic surface of the rough ER is a receptor protein for the SRP. Think of it like as a docking site, if you will. So, if you have this setup where the ribosome, the first part of the protein that's coming off, is bound to that SRP, eventually, as this kind of bebops around the cytoplasm, eventually it'll bump into the surface of the rough ER, and therefore the SRP will hook up with the SRP receptor. And if you'll notice, the SRP receptor is right next door to this large thing here that's called the translocation complex. And if all this works correctly, the SRP docks with the SRP receptor and puts the ribosome right on top of this translocation complex thing. Now this is a tiny bit inaccurate. This is kind of an old drawing. And I'll show you an animation that shows this a little bit better. There is sort of a lid on this translocation complex. Best analogy is those kinds of trash cans where you step on the little pedal thing and the lid goes up. Kind of that idea. So the lid is usually down. Okay, so it sets it on top of it. So there is kind of a lid here in reality. Again, I'll show that to you in the animation. Okay. So what have we done? We've gotten this ribosome that's translating this protein that needs to get into the ER. Well, now we've gotten it to the rough ER. Okay, by binding that SRP to its receptor, we put the ribosome on top of the translocation complex, and then the final thing that the SRP is going to do is sort of thread that initial part of the protein into the translocation complex. Okay? Basically, what this translocation complex is, you might be getting the idea here, is how we're going to get this big protein across the membrane. So it's a tunnel or a channel, whatever we want to think about it. And then once that happens, the SRP has done its job, it leaves and goes back to find another protein that needs to get to the rough ER. And so it's recyclable, which is nice. Does that part make sense? So only proteins that have this special signal, these 16 to 20 hydrophobic amino acids, go through this process. If you don't have that signal, the SRP can't bind, and therefore you don't get to the rough ER. Does that kind of make sense? Okay, so we'll pick up here. So now our protein has been sort of threaded through this translocation complex. The SRP has left, so what can start up again? Translation. Okay, so now the ribosome starts, or continues really, to translate this protein. And now that it's sort of been, sort of threaded through, as the protein elongates, it starts being pushed through the translocation complex. It's kind of clever. So this is how this protein is going to cross from the cytoplasm, across the ER membrane into what's called the ER lumen, okay, the inside of the ER, just like we had like the thylakoid lumen, the endoplasmic reticulum lumen, the inside of the ER. And then there's one last step to all of this. The whole point of this signal was just to get the protein to the ER. So now that it's done that job, it gets snipped off by this final protein here called signal peptidase. And its job is to just snip off the first 16 to 20 hydrophobic amino acids. They did their job. They got the protein to the ER. That's the only job they have. They don't belong in the final protein. So they get snipped off, and the rest of the protein drawn in blue here, in this example, enters the ER lumen. And probably, these first 16 are chopped up again into individual amino acids and are probably reduced, I would imagine. I don't know if we know that for sure, but that seems reasonable. So here's our protein now. Instead of floating around the cytoplasm, now it's floating around inside the rough ER.
You said it only cuts off the first 16 or 20?
Right. Cuts off the signal sequence.
Oh, the signal sequence.
Right, because the signal sequence is the first 16 to 20 hydrophobic amino acids.
You say that in step 2 it starts translation?
It does, because the SRP has left. It was the SRP that was saying, don't translate, don't translate, don't translate, don't translate, don't translate. Now it's gone. It starts translating again. Okay, so this is the essential story so far for 2 of the 3 kinds. So what this little blue protein represents right now is either a secretory protein or a lysosomal enzyme. We can't really tell yet. We have to keep going with the process to figure that out. But this protein right here would represent one of those first 2 types. So we still have to deal with the 3rd type because there's going to be, as we'll see in a second, there's going to be an extra step for the 3rd type. Okay? Because it turns out that if you're a protein that needs to get inserted into the membrane, this is where it's going to happen. You're going to first get inserted into the ER membrane, as it turns out. I'll hold off for that in a second. Okay? Everyone feeling okay with this first part of the story? Okay, so let's deal then with that 3rd part. Oh, actually, this is the same thing. It's just the same picture, just from a different book. So it's showing the same thing. So here's the signal sequence. Here's the SRP. We bind. We stop translating. We hook up with the SRP receptor. That puts the ribosome on top of the translocation complex, inserts it in. Translation picks back up, pushes the protein through. The signal peptidase cuts off the signal sequence. here's our protein inside the ER. So it's showing the same thing, it's just, like I said, a series of pictures from a different book. Here we go. Okay. So, if you're a protein that needs to get inserted into the membrane, the initial part of the story is exactly the same. You have that signal sequence, the first 16 to 20 amino acids, the SRP binds, it gets you to the rough ER. Right, the signal sequence is chopped off. But there must be a difference if this protein, as you can see, if we jump ahead, is going to get inserted. So the difference here, if you're a protein that needs to get inserted into the membrane, guess what? You have a second signal, which kind of makes sense. So the first signal is the same one as we had here, okay, that got you to the ER. But instead of going all the way into the ER, the signal to insert the protein into the membrane is right here. It's called a stop transfer sequence. It's really kind of the same thing as this one. It is a stretch of, it might not be quite as large as that. Maybe it's like 14 to 18 or something like that. But it is a stretch of hydrophobic amino acids again. So it's very similar to the first signal. But instead of being at the end, as you can see here, it's somewhere in the middle of the protein or something like that. And this stop transfer sequence stops translation again, somehow. We don't know exactly how. Okay, again, it doesn't stop it permanently, but just for the moment. And acts as the transfer signal. Somehow, this stretch interacts with the translocation complex and says, insert me into the membrane. As far as I know, we don't know exactly how that happens. But somehow, this translocation complex lets the protein out, inserts it into the membrane, so here's that hydrophobic stretch, which kind of makes sense, right? If we're going to put a protein into the membrane, that section of the protein has got to be hydrophobic, right? Because the lipids of the membrane are hydrophobic, so it kind of makes sense. And then once that's inserted, translation picks up again, and in this example, the rest of the protein then is pointing out into the cytoplasm. So we have part of it in the ER, part of it crossing the ER membrane, and part of it sticking in the cytoplasm. And it turns out that this orientation will be maintained. So if this protein is destined for the cell membrane, so maybe it's a receptor or maybe it's a part of a translocation complex or whatever, the part that's in the cytoplasm stays in the cytoplasm. The part that's now inside the ER is the part that will be pointing outwards, out to the outside of the cell. So that orientation is maintained. So here's our third type of protein that has to go through the ER, that gets inserted into the membrane. By a second signal, these stop transfer sequences inserted into, or somehow interact with the translocation complex, and they end up inserted into the membrane. So once again, we're sort of deceiving yourselves to think we know how everything works inside a cell. We definitely do not. So please, someone go to graduate school, figure this out, so I can stop saying we don't know how this works. I've been saying that for 15 years. I'm tired of saying that. So somebody needs to figure this out. So this is sort of the simplest version, where the protein just crosses the membrane once. Everything else is just sort of a variation on this. So there are proteins that cross the membrane multiple times. We'll come across that in another week or so when we look at cell communication. There are receptors that cross the membrane 7 times. So it's just kind of a repeating of this basic process. If we need this to cross again, there's going to be another signal that gets that part of the protein back into a translocation complex and back out into the membrane. Okay, so we cross again and again. So it's just a variation on this theme. So if you understand how it crosses once, I will be very happy. Anything more complicated than that is just kind of somewhat of a repetition of this basic process. Okay, so there can be multiple stop transfer sequences to get this crossing the membrane multiple times. That's sort of the bottom line. Okay. But like I said, if you understand how it crosses once, I will be ecstatically happy. Again, this is basically the same picture from a different book, also showing that in the first instance we have the amino terminus on the inside of the ER and the carboxy terminus in the cytoplasm. You can rearrange the signals to get the opposite orientation. So in this example, the signals are oriented a little bit differently, and therefore you get the protein oriented differently, where the carboxyl side is in the ER and the amino side is pointing out to the cytoplasm. That's critical as well. So how the signals are placed within the protein is going to determine not only how many times it crosses the membrane, but the orientation of what side of the protein is going to stick to the outside of the cell, what side of the protein is going to be inside the cell. And that's critical. If it needs to be this way, then if you invert this, the protein won't work. Okay, so it's critical that it crosses the right number of times and the orientation is correct. Okay. But again, that's a level of detail I don't think we need to get ourselves bogged down in, but realize that how these various signals are placed along the protein will determine both of those things. So how many times it crosses the membrane and the orientation in terms of the amino and the carboxyl ends of the protein. But like I keep saying, if you understand this basic picture, I will be incredibly, incredibly happy. So how are we feeling about that basic picture? Does the stop transfer sequence kind of make sense? Okay, so that's the initial story then of our big three types of proteins. Right? The secretory type of protein, the lysosomal enzyme, usually just have that initial signal to get to the ER, and they cross entirely into the ER. And now if you're an integral membrane protein, you typically need that second stop transfer sequence. That's what gets you inserted into the membrane. And once you're inserted into the membrane, you're going to stay in the membrane for the rest of the ride, basically. So we've looked at now the ER part of these three, at least the beginning part of the ER story here. Okay. Yeah, let's take a look at that next animation. Okay, so this is going to kind of put what we just looked at kind of in motion. So here's the ribosome reading along this message. Here is the first part of the protein that's coming off. And again, red is going to indicate that signal, that ER signal sequence. Okay, here's our SRP. Looks for that ER signal sequence, binds there. Somehow interacts with the ribosome to stop translation. Now it's going to bump into the SRP receptor on this cytoplasmic side of the rough ER. And there's that lid I told you about. Okay. It's going to place then the ribosome on top of the translocation complex. The little lid opens, threads the protein in. Now it's done its job. It leaves. Translation picks up again. And it pushes the protein through the translocation complex. Here's Pac-Man signal peptidase, cleaves off the signal. Which again is presumably those amino acids are probably used. And here comes the rest of the protein. So again, this would represent either a secretory protein or a lysosomal enzyme. If you need to get inserted into the membrane, your signals are oriented a little bit differently. So here's a sort of more internal signal. That's going to be both the ER signal and a stop transfer sequence. So sometimes one signal can do both jobs, since they're both pretty much the same signal. And then somehow the stop transfer sequence is going to allow the translocation complex to put the membrane, or put the protein into the membrane. And how that exactly happens, I don't think we understand yet. Okay, so here's our integral membrane protein oriented in this example with the amino side in the cytoplasm, the carboxyl side in the ER, and this eventually will be the part that points to the outside of the cell. So as usual, all of these animations are up on the internet. Okay. So far so good. We're good on how the proteins get to the ER and then how some of them get inserted into the membrane. Okay? Okay. There are some other things that are going to happen, or that can happen, to proteins while they're in the ER. So these are usually referred to as post-translational modifications. So we're used to that, right? We have post-transcriptional modifications, right? We have the G-cap and the intron removal and the 3' poly A tail. So here we are after translation, and so we're going to modify proteins. Okay? So these are collectively known as post-translational modifications. ER. So there are 2 or 3 that we'll talk about. The first one that you see here is that some proteins are going to pick up some sugar. And that process is called glycosylation. Now, some proteins pick up sugars and some don't. So what do you think is going to be involved? A signal. Okay? So there is apparently a special signal for proteins to pick up some sugars. And that signal is, there we go. It's a 3 amino acid signal, an asparagine, X meaning any amino acid, and then the third one is either a threonine or a serine. So it's asparagine, X, threonine, or serine. It is the signal that, hey, this protein needs to get some sugars. And again, we know that in the same way we know about all kinds of other signals. If you take that off of a protein, guess what? It doesn't get any sugar. If you add that to a protein that usually does not get sugar, guess what? You get sugar. So we're pretty confident that that's the signal. So this is kind of how that works. At least initially, there's sort of a prefabricated set of sugars here. Okay, so some N-acetylglucosamines, some mannoses, some glucoses. This is all sort of prefabricated. And it's all initially attached to a special lipid in the membrane called dolichol. So it's kind of pre-manufactured. It's just kind of sitting there waiting for a protein with that signal. And hey, here comes a protein with that signal, an asparagine, X, threonine, or serine. And what happens is that an enzyme will take the sugars off of the lipid, off the dolichol, and attach it to the asparagine. Okay, so the sugars get attached to the first amino acid here, the asparagine. So that has been well worked out. What is less worked out is the rest of the picture here. Other enzymes seem to modify this sort of prefabricated set of sugars. So in this example, other enzymes will, in this example, cut off the 3 glucoses and cut off that particular mannose. Now, why? I don't know. There obviously must be other signals involved that somehow tell other enzymes how to sort of modify this sort of what's usually called the core oligosaccharide. But we certainly don't understand those other signals yet. Because what we do know is that after they pick up this sort of prefabricated thing, it is often modified, and modified in different ways for different proteins. So presumably there have to be other signals that are sort of directing that, but those signals have not been worked out yet. So lots of stuff still to figure out about the rough ER. So if you're interested in the rough ER, there are, like I said, still a lot of questions to be answered. So we know part of the story. Okay, so the big section of carbohydrates gets attached to a protein if it has that 3 amino acid signal and gets attached to the asparagine amino acid specifically. So lots of proteins pick up sugars. Okay, so that's one big post-translational modification. Yes, sir.
You said the dolichol was a lipid?
It is. Well, it's a lipid alcohol, if you want to be perfectly accurate. Okay, so that's our first one. They pick up sugar, or many of them pick up sugar, but not all. So if you don't have the asparagine X-threonine signal, you don't pick up any sugar at all. So you can run the gamut from no sugar to lots of sugar. Okay, but glycosylation in general. Oh, we're on time. Okay, so we'll stop here. So we have, I think, 2 more modifications to go in the ER, and then we'll travel from the ER to the Golden. Okay, so we'll work on that on Monday. Please, my dear friends, start studying this.